For decades, there was a quiet, almost universally accepted assumption running through the entire field of ancient human genetics. DNA simply does not survive long enough to be useful once you go back much further than about 100,000 years. Organic molecules break down. Bone, mineral degrades. Groundwater, temperature fluctuations, and the sheer relentless passage of geological time destroy the fragile genetic material of ancient organisms long before it can tell scientists anything meaningful. This assumption shaped what questions researchers even considered worth, attempting to ask about the deep human past, and it quietly closed off huge stretches of the human evolutionary record to genetic investigation, leaving scientists to rely on bone shape and stone tools alone to reconstruct what happened during the hundreds of thousands of years before that horizon. Then, in a cold, isolated cave system in the mountains of northern Spain, a group of researchers extracted genetic material from human remains that were somewhere in the range of 400,000 years old, an achievement that shattered the assumed limits of ancient DNA recovery. And when they finally sequenced it, the result was not simply a triumph of laboratory technique. It was a genuine scientific puzzle, one that did not match the expected genetic profile of the population these bones were assumed to belong to, and that instead pointed toward a completely different and almost entirely unexpected branch of the human family tree. The bones looked like one kind of ancient human. The DNA said something else, and reconciling that contradiction would take scientists years, an entirely new round of even more difficult extraction work, and a rethinking of how tangled the deep roots of the human story actually are. The site responsible for this discovery is called Sima de los Huesos, which translates roughly from Spanish as the Pit of Bones, a name that is disarmingly literal. It sits deep within the Atapuerca cave system in the Sierra de Atapuerca mountains of northern Spain, at the bottom of a nearly vertical shaft that drops some 13 meters through solid rock before opening into a small chamber. Reaching the site today requires crawling, climbing, and descending through narrow twisting cave passages using ropes and headlamps. And it is difficult to overstate how strange it is that any group of ancient hominins would have ended up there at all, in a location that offers no natural light, no easy entrance, and nothing resembling a habitable living space. That strangeness is, in fact, part of what makes the site so scientifically important. Since serious excavation began in the 1980s under the direction of Spanish paleoanthropologist Juan Luis Arzuaga, building on earlier survey work led by Emiliano Aguirre, researchers have recovered the skeletal remains of what is now estimated to be at least 28 separate individuals from the chamber at the bottom of the shaft, making Sima de los Huesos by a wide margin the largest single collection of Middle Pleistocene hominin fossils ever found, anywhere in the world. Skulls, jawbones, vertebrae, pelvises, and thousands of smaller bone fragments have been painstakingly recovered, cataloged, and reassembled over more than 3 decades of continuous fieldwork, producing an assemblage so extensive that it has effectively rewritten what scientists thought they knew about the population of early humans living in Europe, several 100,000 years before the emergence of anatomically modern humans. How so many individuals ended up at the bottom of a vertical shaft that appears never to have been used as ordinary living space remains one of the site's genuine open questions. Arzuaga and his colleagues have proposed that the accumulation represents deliberate, repeated disposal of the dead by the hominin population that produced them, effectively a very early form of intentional mortuary behavior, since there is no evidence the individuals fell in accidentally over time or were dragged there by carnivores, whose remains are notably rare at the site by comparison. If this interpretation is correct, and it remains debated, Sima de los Huesos may represent one of the oldest examples of deliberate human treatment of the dead found anywhere on Earth, a claim that carries its own significant weight independent of anything the genetic material would eventually reveal. Establishing exactly how old the Sima de los Huesos remains actually are has itself been a complex, multi-decade scientific undertaking, involving several independent dating methods applied to the sediments and the bones themselves. Uranium series dating, which measures the radioactive decay of uranium isotopes trapped in mineral deposits, combined with electron spin resonance dating, which measures radiation damage accumulated in tooth enamel and other crystalline materials over time, converged on an age for the deposit of approximately 430,000 years, placing the individuals firmly within the Middle Pleistocene, a period of human evolution so deep in the past that it predates the emergence of both Neanderthals and modern humans as fully distinct, recognizable populations. This dating placed the Sima de los Huesos population at a genuinely pivotal moment in human evolutionary history, sitting close to the period when the lineage that would eventually produce Neanderthals in Europe and the lineage that would eventually produce modern humans in Africa were still diverging from their shared ancestral population, or had only recently done so. Skeletal analysis of the recovered bones showed a suite of features, including the shape of the brow ridge, characteristics of the inner ear bones, and details of dental morphology that pointed consistently toward an early primitive form of the Neanderthal lineage specifically, rather than toward the ancestors of modern humans, or toward some entirely separate and unrelated population. Based on the bones alone, in other words, the scientific expectation was clear and for years largely unquestioned. These were early Neanderthals, or something very close to the population that would eventually give rise to Neanderthals across Ice Age Europe. For a long time, the question of what these individuals' genetic material might reveal remained purely theoretical, because nobody seriously believed DNA could survive intact in bone that old, especially bone that had spent 430 millennia in a cave. Environment, however cold and stable that environment might have been compared to more exposed sites, ancient DNA is extraordinarily fragile. It degrades into shorter and shorter fragments over time, becomes chemically damaged in ways that can introduce errors into sequencing, and is almost always overwhelmingly contaminated by DNA from soil bacteria, fungi, and other environmental sources that vastly outnumber whatever trace amounts of hominin genetic material might remain. The team that eventually took on this challenge was led by the paleogeneticist Matthias Meyer, working within the research group directed by Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, a laboratory that had already established itself as the world's leading center for ancient hominin genetics through its earlier work sequencing the Neanderthal genome and identifying the previously unknown Denisovan population from genetic material recovered in a Siberian cave. Even against that backdrop of established expertise, attempting to recover usable genetic material from bone as old as the Sima de los Huesos remains was considered a long shot bordering on the technically implausible, since it would require pushing the recovery of ancient DNA roughly 3 to 4 times further back in time than had ever been reliably achieved before. In 2013, Meyer and his colleagues announced that they had succeeded, at least partially. Working with a fragment of thigh bone from the Sima de los Huesos assemblage, the team managed to extract and sequence a complete mitochondrial genome, the small, separate loop of DNA that exists outside a cell's main nucleus and is inherited exclusively along the maternal line. Mitochondrial DNA exists in far higher copy numbers per cell than nuclear DNA, which made it a more realistic target for recovery from remains this ancient, even though it represents only a tiny fraction of an organism's total genetic information, and is inherited along only one line of descent rather than reflecting a full genetic picture of ancestry. Given everything the skeletal evidence at Sima de los Huesos had already indicated, the expectation going into this analysis was straightforward. If the mitochondrial genome could be recovered and compared against the known genetic profiles of Neanderthals, modern humans, and the more recently identified Denisovans, it should show the individual to be most closely related to Neanderthals, or to whatever ancestral population directly preceded them, consistent with everything the bones themselves appeared to say. Instead, when the sequence was compared against the existing reference genomes, it showed a genetic relationship that was closer to the Denisovans, the archaic human population first identified in 2010 from a small fingerbone fragment and a molar tooth recovered from Denisova Cave in the Altai, mountains of Siberia, thousands of kilometers to the east of Spain. This was, by any measure, a startling result. The Sima de los Huesos individual looked, by every measurable skeletal criterion, like an early Neanderthal. Its mitochondrial DNA, however, told a story that pointed instead toward a genetic connection with a population whose only other known physical remains had been found on the other side of the Asian continent, in an environment and a time period that did not obviously overlap with conditions in Ice Age Spain. The finding did not fit comfortably into any of the existing models of how the human family tree was organized. It raised the immediate and difficult question of how a population living in Spain, exhibiting Neanderthal-like skeletal anatomy, could carry a maternal genetic lineage more closely related to a population known only from remains found in Siberia. Several possible explanations presented themselves, none of them simple, and none of them capable of being fully resolved on the strength of a single mitochondrial genome alone. It was possible that the population ancestral to both Neanderthals and Denisovans had been more genetically diverse and more geographically widespread than previously understood, with the Sima de los Huesos individuals representing an early branch that retained a maternal lineage later associated primarily with the eastern Denisovan population. It was possible that some earlier unknown episode of interbreeding had introduced a Denisovan-related mitochondrial lineage into the Sima de los Huesos population from an entirely separate, and at that time still completely unidentified, archaic human group. And it was possible, though this explanation gained less traction among specialists, that the skeletal classification of the Sima de los Huesos remains as early Neanderthals needed to be reconsidered altogether. Because a single mitochondrial genome inherited only along the maternal line can never tell the whole story of a population's ancestry, The Meyer and Pabo team pushed forward with an even more technically demanding project, attempting to recover nuclear DNA, the genetic material contained within a cell's nucleus that reflects contributions from both parents and offers a vastly more complete picture of ancestry and population relationships than mitochondrial DNA alone ever could. Nuclear DNA exists in far lower copy numbers per cell, making it, correspondingly, more difficult to recover from degraded ancient material, and the technical challenge of extracting usable nuclear sequences from bone that was, by this point, established at roughly 430,000 years old, was considerably greater than the challenge that had already produced the surprising mitochondrial result. In 2016, the team published the results of this expanded effort in the journal Nature, reporting that they had successfully recovered nuclear genome sequences from several individuals at the Sima de los Huesos site. This time, the results aligned far more closely with what the skeletal evidence had suggested from the beginning. The nuclear DNA showed that the Sima de los Huesos population was indeed more closely related to Neanderthals than to Denisovans or to modern humans, confirming the population's position as an early ancestral branch of the Neanderthal lineage, essentially resolving the apparent contradiction that the earlier mitochondrial result had created. But resolving that contradiction did not make it disappear so much as relocate it. If the nuclear DNA confirmed a primarily Neanderthal-related ancestry, then the earlier finding of Denisovan-related mitochondrial DNA needed its own explanation, since the two results taken together could not both simply reflect a single, straightforward population history. The most widely discussed explanation to emerge from the research team and the broader community of paleogeneticists who examined the findings afterward is that the mitochondrial año mainly likely reflects an ancient episode of interbreeding, in which a maternal lineage from a population related to, or ancestral to, the later Denisovans was introduced into the population that would go on to become the Neanderthal lineage, replacing whatever mitochondrial lineage that population might otherwise have carried, while the nuclear genome, reflecting contributions from many more ancestors across many more generations, retained its dominant, recognizably proto-Neanderthal character. What this combination of results points toward is a picture of deep human ancestry considerably more tangled, more regionally fluid, and more prone to episodes of interbreeding between distinct archaic populations than the relatively clean, tree-like model that dominated earlier decades of paleoanthropological thinking. For much of the 20th century, human evolution was often visualized as a single branching tree, with populations splitting cleanly from common ancestors and then evolving in isolation from one another, until they either went extinct or gave rise to further distinct branches. The genetic evidence recovered from ancient hominin remains over roughly the past 15 years, and Sima de los Huesos represents one of the most striking examples of this trend, has consistently pushed against that clean model, replacing it with something closer to a tangled, overlapping network in which distinct archaic populations periodically encountered one another, interbred, and exchanged genetic material across geographic distances and time depths that earlier researchers would not have thought plausible. The Denisovans themselves are a particularly useful illustration of just how much this picture has changed. Before 2010, no scientist anywhere had proposed the existence of a distinct archaic human population east of the Neanderthal range in Ice Age Asia because there was no fossil or genetic evidence pointing toward one. The identification of the Denisovans rested almost entirely on genetic analysis of a tiny fingerbone fragment, so physically nondescript that it would never have been flagged as anything unusual on skeletal grounds alone, and yet the DNA extracted from that fragment revealed an entire, previously unknown branch of the human family tree, one that subsequent research has shown interbred not only with the ancestors of some modern human populations, particularly in parts of Asia and Oceania, but potentially, as the Sima de los Huesos mitochondrial result suggests, with early European populations as well, at a time depth far earlier than anyone had previously considered plausible. The Sima de los Huesos discovery matters for reasons that extend well beyond resolving the specific technical puzzle of one anomalous mitochondrial genome. It matters first simply as a demonstration of what is now technically achievable in the recovery of ancient genetic material since the successful extraction of both mitochondrial and nuclear DNA from remains approaching 500,000 years in age fundamentally shifted the assumed outer limit of what ancient DNA research could accomplish, opening the door to genetic investigation of hominin populations that had previously been considered permanently inaccessible to anything other than skeletal and archaeological analysis. It matters, second, because of what it reveals about the genuine complexity of the deep human past, a complexity that skeletal analysis alone, however careful and rigorous, could never have fully uncovered. The population at Sima de los Huesos looked like early Neanderthals because, according to the fuller nuclear evidence, they largely were early Neanderthals. But their maternal genetic history carried a trace of something else entirely, a hint of contact or shared ancestry with a population whose descendants would eventually turn up on the opposite side of the Asian continent tens of thousands of years later, known to modern science only through a single finger bone in a Siberian cave. That is not the kind of detail that bone shape, tooth wear, or stone tool typology could ever have revealed on its own. It required going inside the bones themselves, down to the level of the genetic code, to find it. And it matters, finally, because of what it suggests about how much more remains to be discovered as ancient DNA recovery techniques continue to improve. If a population living in a cave in northern Spain more than 400,000 years ago carried genetic traces connecting it to a population identified from remains found thousands of kilometers away, the deep human past likely contains a great many more such connections, encounters, and exchanges that current evidence has not yet uncovered, simply because the genetic material needed to reveal them has not yet been successfully recovered or has not yet been recovered from the specific individuals and populations where those connections would be visible. The scientific community has not settled every question raised by the Sima de los Huesos genetic findings, and researchers continue to debate several aspects of what the results actually mean. The precise nature and timing of whatever ancient interbreeding event produced the Denisovan-related mitochondrial lineage found in the Sima de los Huesos population remains uncertain, since the current evidence cannot fully specify which population contributed that lineage when the exchange occurred, or how geographically widespread the population carrying it might have been at the time. There's also ongoing discussion about how representative the handful of individuals from whom nuclear DNA has so far been successfully recovered actually are of the broader Sima de los Huesos population as a whole, given that the site contains skeletal material from at least 28 individuals. But genetic data has only been recovered from a small subset of that total, raising the possibility that genetic diversity within the population itself, including further unexpected mitochondrial lineages, may yet be revealed as sequencing efforts continue and as extraction techniques become more sensitive to smaller quantities of increasingly degraded genetic material. And the broader theoretical question of how to properly model human evolution during this period, given mounting genetic evidence of repeated interbreeding between distinct archaic populations across wide geographic and temporal distances, remains an active and unsettled area of paleogenetic research, one that the Sima de los Huesos findings contributed to significantly, but by no means resolved on their own. Recovering genetic material from bone this old is not simply a matter of grinding up a sample and running it through a sequencer. Ancient DNA labs like the one Paabo established in Leipzig operate under extraordinarily strict contamination controls, because the DNA researchers are trying to recover exists in such vanishingly small quantities and in such a degraded state that even minute amounts of modern human DNA, whether from a researcher's skin cells, breath, or earlier handling of the sample by museum staff decades before, can overwhelm and contaminate the signal researchers are actually trying to detect. Samples are processed in dedicated clean rooms, physically separated from any area where modern DNA samples are handled, using positive air pressure systems, full protective clothing, and surfaces regularly treated with bleach and ultraviolet light to destroy any stray genetic material. Researchers drill small amounts of bone powder from the sample, usually from the dense protective petrous portion of the skull's temporal bone when it is available, since this particular bone has repeatedly proven to preserve genetic material unusually well compared to other skeletal elements, likely because of its extremely high density and correspondingly low porosity to water and microbial infiltration over long periods of burial. Once extracted, the DNA fragments recovered from samples of this age are typically extremely short, often only a few dozen base pairs long, chemically damaged in patterns that are themselves diagnostic of genuine ancient origin, as opposed to modern contamination, and vastly outnumbered by DNA from soil bacteria and fungi that colonized the bone over hundreds of thousands of years of burial. Reconstructing a usable genome from this kind of degraded starting material requires sequencing enormous numbers of these short fragments, and then using computational methods to align them against reference genomes from Neanderthals, Denisovans, and modern humans, distinguishing genuine ancient hominin sequence from environmental contamination through statistical patterns in fragment length and characteristic chemical damage that accumulates predictably over time. It is a slow, exacting, expensive process, and the fact that it succeeded at all on material approaching 500,000 years old was, even to specialists in the field, a genuinely surprising technical achievement. When the initial 2013 mitochondrial result was first presented at scientific conferences and subsequently published, it was met with a considerable amount of professional caution, and in some quarters outright skepticism, from researchers who had spent careers studying both the Sima de los Huesos fossils and the broader question of Neanderthal and Denisovan population relationships. Extraordinary claims of this kind, particularly ones involving DNA recovery from remains far older than anything previously achieved, invite intense scrutiny, and several independent research groups work to evaluate whether the reported sequence could plausibly be explained by contamination, sequencing artifacts, or misidentification rather than genuine ancient signal. The characteristic chemical damage patterns present in the recovered sequence, consistent with genuine ancient DNA rather than modern contamination, alongside the internal consistency of the results across multiple extraction attempts from different bone samples, ultimately persuaded the broader research community that the finding was real, even as the explanation for what it meant remained unsettled. This pattern, in which an ancient DNA result is first treated with considerable institutional caution before being gradually accepted as further evidence accumulates, has repeated itself throughout the short history of the field. And it reflects an appropriately high evidentiary bar for a type of finding that is both technically difficult to produce and unusually easy to get wrong. The Sima de los Huesos results did not emerge in isolation. They form part of a broader body of work that has, over roughly the past 2 decades, repeatedly forced paleoanthropologists and geneticists to revise their expectations about both the technical limits of ancient DNA recovery and the underlying structure of the human family tree itself. Pabo's laboratory had already achieved a series of milestones that once seemed similarly implausible, including the sequencing of a complete Neanderthal genome in 2010 from remains found in Croatia, and the identification of the entirely unknown Denisovan population that same year from the Siberian fingerbone fragment, work that was recognized in 2022 when Pabo was awarded the Nobel Prize in Physiology or Medicine for his contributions to the field of paleogenomics. Each of these achievements shared a common pattern with the Sima de los Huesos discovery, a technical breakthrough in DNA recovery that produced results considerably more complex and more genuinely surprising than the prevailing scientific expectations had anticipated. The Neanderthal genome revealed that Neanderthals had interbred with the ancestors of modern humans outside Africa, a finding that overturned decades of assumption that Neanderthals had contributed nothing to the modern human gene pool. The Denisovan discovery revealed an entire previously unknown branch of the human family tree, identified from remains so fragmentary that no one had previously suspected its existence. And the Sima de los Huesos mitochondrial anomaly revealed that the population relationships among these archaic human groups were considerably more tangled and involved considerably more ancient interbreeding than even the already revised post-Neanderthal genome picture of human evolution had assumed. Fieldwork at Sima de los Huesos has not stopped. Juan Luis Arzuaga and his research team continue to excavate the site during regular summer field seasons, and the total number of recovered skeletal elements has continued to grow year over year, along with the number of individual hominins represented in the assemblage. Each new season raises the possibility that additional bone fragments suitable for genetic analysis, ideally including well-preserved petrous temporal bones, will be recovered, potentially allowing researchers to build out a genetic picture of the population's diversity considerably beyond the handful of individuals from whom nuclear DNA has so far been successfully sequenced. That kind of expanded sampling matters enormously for resolving the remaining open questions about the population's genetic history, since a larger number of individually sequenced genomes would allow researchers to determine how widespread the Denisovan-related mitochondrial lineage actually was within the Sima de los Huesos population, whether it appeared in some individuals, but not others, and whether additional, still undetected genetic surprises might be waiting in bone fragments that have not yet been sampled, or that have not yet even been excavated from the sediment at the bottom of that narrow vertical shaft. What makes the story of the Sima de los Huesos DNA genuinely remarkable is not that scientists found something that defies explanation entirely. It is that they found something that, for a period of real scientific uncertainty, appeared to contradict everything the physical remains themselves seemed to indicate, and that resolving that contradiction required not simply more careful interpretation of existing evidence, but an entirely new round of even more difficult laboratory work, extracting nuclear genetic material from bone that had already been considered barely at the edge of what ancient DNA recovery could achieve. The bones at the bottom of that vertical shaft in the Atapuerca Mountains looked, by every conventional measure, available to paleoanthropologists, like early Neanderthals, and the fuller genetic evidence has ultimately confirmed that basic classification. But buried within that expected Neanderthal ancestry was a genuine surprise, a maternal genetic. Thread connecting a population in Ice Age Spain to another archaic human population, identified from a single fingerbone on the far side of Asia, a connection that nobody examining the skeletal remains alone would ever have had reason to suspect. It is a reminder that the deepest layers of the human story are still being written, not primarily in the shape of ancient bones, but in the genetic code recovered from inside them, one increasingly difficult extraction at a time.